everyone. The title of our presentation is the in-silico characterization of epigenetic drugs for epigenetic targets such as DNA Methyltransferase and histone deacetylases on the HCT116 colorectal cancer cell line. And I'm Shikha. For a brief overview of our presentation, I'm going to first talk about the purpose and background, then I'll dive into the methods and the tools we used, and highlight some of our current progress. Finally, I'll explain the conclusion and some future steps and references. According to the American Cancer Society in 2022, colorectal cancer is the third leading cause of cancer-related deaths, and 52,580 deaths are expected from colorectal cancer this year. When expressed, DNMTs and HDACs lead to cancer growth by altering the expression of tumor suppressor genes. Thus, the purpose of our project is to determine novel DNMT and HDAC inhibitors for colorectal cancer. So what really are DNMTs? DNMTs are responsible for maintaining methylation patterns in the cytosine guanine regions of DNA. DNMTs add methyl groups, altering the epigenetics of DNA, making some parts of it heterochromatin. This tighter, condensed chromatin is harder to transcribe, and the genes within it are silenced. When these genes are tumor suppressor genes, the result is cancer growth. In our group's research, the main focus is DNMTs, because DNMT3 sets up organisms' methylation patterns before birth. The image on the right was made using pre-existing software tools known as PyMol and P2Rank. Cheminformatics tools used to showcase the binding pockets in DNMT1, where a potential inhibitor could bind. The other group of proteins we are focusing on is HDACs, or histone deacetylases. As the name suggests, it removes an acetyl group from the lysine on specific types of proteins. HDACs also make the DNA less accessible to transcription factors, and in colorectal cancer prevents the function of tumor suppressor genes. This diagram shows an overview of our workflow. First, we examined the drug databases such as PubChem or Chembl, and after finding our data, we divided it into chunks of 1,000 compounds to perform computational analyses. Then we clustered them using techniques such as t-SNE and UMAP, and the candidates most similar to existing DNMT and HDAC inhibitors will be screened using molecular docking tools such as AutoDocVina. Our next steps in the methods include to use molecular simulation tools like GROMACS to better examine drug-protein binding, using deep CDR to simulate how inhibitors would function on the HCT116 colorectal cancer cell lines, and after finding the most optimal drug, our last step would be to test for drug-induced liver injury potential. In order to complete the methods detailed in the last slide, we are using several tools. I'd like to clarify that although we are using these tools, they were created by someone else not in our lab. The citations for all the tools will be listed in the last slide and in our references document. After examining databases of compounds including PubChem and Chembl, we ultimately chose Chembl because even though it had less compounds, it was manually curated and we were able to bulk download all the data in a CSV file. Then, we modified the dataset in Excel by adding a column of ECFP fingerprints to the molecules, which was generated by using RDKit Cheminformatics library in Python. We then clustered the data using t-SNE and UMAP, which helped us determine drugs that were most similar to existing DNMT inhibitors, and were likely to exhibit inhibitory properties as well. After determining potential novel inhibitors, we used Avogadro to visualize the compound and Orca to optimize the geometry of potential inhibitors. We also plan on using AutoDocVina to determine the ligand binding affinity of the inhibitors to DNMT and HDAC. To filter for false positive binders, we'll use molecular simulation tools such as GROMACS or OpenMM. These tools vibrate the individual atom of the protein and the individual atom of the drug to see how well they bind. Then we plan to use TorchMD to determine a biomolecular system response to disturbance. The force computations, including Coulomb interactions, are expressed as PyTorch operations. TorchMD is unique in that it also integrates neural network potentials into its predictions. Next, we plan to use DeepCDR. DeepCDR predicts drug sensitivity in two ways. First, it gives the binary value of sensitivity, and second, it gives the IC50, which is the half maximal inhibitory concentration, which is the concentration required to inhibit the growth of 50% of the cells. Lastly, to make sure that our patient is safe, we will use an open-source convolutional neural network made by the Bin Pien Nguyen lab. The graph on this slide shows the current progress from the clustering step. We used some known DNMT inhibitors and clustered 1,000 compounds to see which ones were the most similar to the existing DNMT inhibitors. In this graph, the known inhibitors are colored in blue, whereas the other compounds are orange. We plan to continue working on our project, and once we complete in-silico screening of the drugs, the wet lab group in the Kanha lab will test them on colon cancer cells. In the future, we also plan to look at protein signatures in sharks to determine whether the proteins that increase stability in shark cells can be used to increase the stability of human genomes against cancer. We also want to explore other protein targets within cancer and explore neurological disease proteins. Since we consistently use certain tools on several projects, we're also eager to create an efficient pipeline using SnakeMake or workflow development language to increase the overall lab efficiency. These are some of our key references with the longer list of all our references that you can access by scanning the QR code. Lastly, I'd like to thank some people who this project wouldn't have been possible without. So thank you to the Aspiring Scholars Directed Research Program, the Olive Children Foundation, our mentor, Mr. Kanha, and my teammates Akshati and Ashley, and the NJOO group for helping us with docking, and Mr. Robert Downing for helping us overcome software technical difficulties. I'd also like to thank the ISMB conference for giving us a platform to present the research.